Hello and welcome to Dear Doctor, the channel where we explore the latest and most fascinating medical topics, and today we're going to talk about a new discovery that could help us better understand brain inflammation and its role in various neurological and psychiatric diseases. Brain inflammation, or neuroinflammation, is a natural defensive reaction initiated by the immune system. It enables our cells to fight off aggression, such as injury or infection. But sometimes, this response can get out of control and lead to the onset of serious pathologies, such as Alzheimer's disease, amyotrophic lateral sclerosis, multiple sclerosis, schizophrenia, bipolar disorder, and depression. In the brain, microglial cells play an important role in inflammation and its potential overactivation. They can be activated when dysfunction occurs, phagocytize pathological cells or proteins, and even produce protective substances. But how can we measure and visualize these cells non-invasively and in vivo? Currently, there is only one marker that can be used for this purpose, the TSPO protein, which is present in microglial cells. This protein can be observed by positron emission tomography, PET, a common imaging technique that uses radioactive tracers to detect molecular changes in the body. Hundreds of studies have used PET scans of this protein to explore and quantify microglia. However, no study has succeeded in precisely interpreting the significance of its quantity in the context of an inflammatory reaction. Does a large quantity of TSPO correspond to a large quantity of inflammatory cells? Is it a sign of their overactivation? A team of researchers from the University of Geneva, UNIG, the University Hospitals of Geneva, HUG, Imperial College London, and Amsterdam UMC investigated this question using mouse models of Alzheimer's disease, amyotrophic lateral sclerosis, multiple sclerosis, and post-mortem brain samples from patients affected by the same diseases. They found that a high density of TSPO protein indicates a high number of microglia, but not necessarily their overactivation. In fact, they discovered that TSPO expression is regulated by two different mechanisms, one that depends on inflammation and one that does not. This means that TSPO is not a reliable indicator of the intensity or severity of neuroinflammation. Rather, it reflects the presence and density of microglial cells in the brain. This finding has important implications for the interpretation of previous and future studies using TSPO as a marker for neuroinflammation. It also opens new avenues for developing more specific and sensitive markers that can reveal the functional state and activity of microglial cells. This discovery could help us better understand the mechanisms of neurological and psychiatric diseases that involve neuroinflammation and improve their diagnosis and treatment. That's all for today's episode of Dear Doctor. I hope you enjoyed it and learned something new. If you did, please give this video a thumbs up and subscribe to our channel for more medical topics. And don't forget to leave your comments and questions below. I'll see you next time on Dear Doctor.